Hello, I'm Dr. Michael Fossel. I want to talk to you about aging and age-related diseases and the prospects actually for curing and preventing age-related diseases. It's a remarkable topic, but I think we can get into it in some detail. The, the whole idea of aging has fascinated me for about 50 years. I originally was getting my PhD in neurobiology and my MD at Stanford University, shown here, about 45 years ago. And at that time, my focus really was on the development of the nervous system, which is a remarkable thing to look at. But I found that when I talked to people about the other end of the life spectrum, that is age-related diseases, and particularly aging in the nervous system, people would invariably shrug and say, well, that just happens. I find that an unsatisfactory answer. To me, it denotes an intellectual laziness. And I often find that when people say things just happen, what they mean is they're not looking at details, and things are almost invariably more complex than they realize. So I began to be fascinated by the aging process itself and how it occurs at a most fundamental level, because again, we generally don't actually think about it very carefully. And if you think about historical precedents, what you find is that while we have made enormous inroads in changing the mean lifespan of the human race over the past several hundred years, going from perhaps a quarter of a century to a half century to currently about 3/4 of a century in mean lifespan, we've had no impact on the maximum lifespan, which is estimated at about 120 years. Now, I do not want to focus on the quantity of life, that is the length of the human lifespan. Marcus Aurelius, back 2,000 years ago, pointed out that it's not the length of your life that matters. It really is what you do with it. It is the quality of life. And that's certainly true for me from a medical perspective. I'm not concerned with how long you live, but what we can do to prevent disease, fear, pain, tragedy, the sort of things that destroy people's very souls. So the question that I'm asking really is, what can we do to improve the quality of life with regard to aging and age-related diseases? When I think about the quality of life from an historical perspective, I look back and I ask myself, really, what has medicine done for us? And I find that in general, the biggest impact has been what I think of as conceptual revolutions. For example, microbial theory. It has had a much bigger impact on the quality of our lives. For example, hand washing in delivering babies drastically lowered the maternal and the neonatal death rates. But it also improved the quality of life, as did things like rabies vaccinations, smallpox vaccinations, the first antibiotics, polio immunizations. These are things that had enormous impact, not just on extending our lifespan, but improving the quality of life. And curiously enough, they also actually lowered the cost of medical care. Now, this is very much at variance with what I think of as technical innovations. For example, the latest statin drug or robotic surgery or cardiac transplants, all of which have had some marginal impact on the quality of life, but also have increased the cost of medical care. Now compare that to the conceptual revolution that's signified by microbial theory. For example, in about 1950, if you look at the literature, you find that people worldwide were concerned about the cost of treating polio victims. That is the cost of iron lungs and rehabilitation and braces, nursing care. And there were actually estimates that the healthcare budgets of many developed nations would be bankrupt by the year 2000 as a result of treating polio. That did not happen. And the reason is that along about 1954, we had the very first Salk vaccine, followed shortly thereafter by the Sabin oral vaccine. And currently, far from bankrupting healthcare systems worldwide, the cost of treating polio is estimated by the WHO at about 10 cents a head. So that has had an enormous impact on the quality of human life. Again, these are not technical advances, really. These are advances that are incumbent upon a different way of looking at medicine, a different way of looking at, in this case, at disease in terms of microbial theory. And I suggest we're about to do the same thing with regard to age-related diseases. If we truly can understand the aging process at the genetic and cellular level, I think we'll find that not only can we prevent and cure age-related diseases and thereby drastically improve the quality of human life, but in so doing, we will actually lower, not raise, the cost of medical care. I think that we're in for quite a ride as we improve the quality of life and lower the cost of medical care personally, nationally, and globally, just as we did with regard to microbial diseases of microbial origin. Now, to put this in perspective, let me introduce you to a friend of mine, this is Dr. Leonard Hayflick. He was the first person who, about now 60 years ago, first described cell aging. to the world. And he and I were having a conversation here in late 2019, and he pointed this out. And he's right. I find that whenever we talk about age-related diseases, everyone, in fact, probably everyone listening to this, will agree that the single major risk factor for age-related diseases is aging. It's certainly the best predictive risk factor. But at that point, people, although they tip their hat and agree that aging is important, completely ignore aging altogether, and then move on to symptomatic and component therapies for aging. That is, instead of dealing with the aging process, they deal with things like beta amyloid or cholesterol plaque or degenerative changes in knee joints and so forth. And they no longer ask themselves what aging has to do with it. So again, they agree verbally, but their behavior speaks volumes to the country. So the real issue is what is going on with aging. And one of the problems, and it's a very human one, that we all make is that we have the assumption that we understand something, for example, the aging process. This is something that happened medically. For example, at the beginning of my medical career, back in 1980, as an example, we all agreed that ulcer disease was caused by increased acid and stress. That was a very common understanding, the assumption of understanding. And it wasn't that we in medicine back in 1980 were ignorant or stupid or illogical. It was simply that we didn't look any further. Until about 1983, when these Australian physicians, Robin Warren and Barry Marshall, published the first paper in the British Medical Journal, for which they won a Nobel Prize in 2005, pointing out that in all cases we look carefully, we find that ulcer disease, particularly pyloric ulcer, is caused not by stress and acid, but the underlying fundamental cause is bacteria, in this case, Helicobacter pylori, shown here on the right. But this assumption of understanding is what impedes progress. We, back, as I say, in my beginning of my career 50 years ago, we tended to assume we understand aging. We still do. Here in 1980, we had the assumption that we understood ulcer disease. We did not. And the same thing occurs again and again. Here is a literary example. This comes from a play by Shakespeare. Most of you have heard of this hill. This is Dunsinane Hill. And in the very first act of Macbeth, Shakespeare tells Macbeth, the three witches actually tell Macbeth, and Shakespeare tells us that in fact Macbeth can never be conquered unless Great Burnham Wood comes to this hill, High Dunsinane Hill. And we all make the assumption that we have understood what the witches have told us, and therefore that Macbeth can't be conquered, which is true until Malcolm cuts down Burnham Wood, uses it to shield his soldiers, marches on Dunsinane Hill, and kills Macbeth. Well, it's a very common literary trope. And in fact, Macbeth uses it multiple times in Macbeth and uses it elsewhere too. He leaves the audience with the assumption of understanding that he later then disproves and pulls the rug out from underneath us. But we do the same thing in science, for example. This is a quote from Lord Kelvin, the world's leading physicist probably in 1895. Now, Lord Kelvin knew a great deal about weight and a great deal about air pressure, not very much about aerodynamics. And in consequence, he made this statement about flying machines being impossible, which was disproved by the Wright brothers some 8 years later at Kitty Hawk. Now, their first flight was only about 12 seconds, but it was a remarkable flight. It took most of the world about 5 years before people actually understood that they had accomplished it. And again, the problem was not that Lord Kelvin was ignorant or illogical. The problem was that he made the assumption that he understood what was going on and didn't look any further as regards heavier than air flying machines. We did the same thing a few years later with regard to classical physics. We made the assumption that because we understood things like billiard balls classically, or for that matter, computer mice, that we therefore understood atomic and subatomic particles. That is, atoms and subatomic particles like electrons, protons, and neutrons, which in fact are probably not particles at all. But it wasn't until 1924, publication of the first paper on quantum mechanics, and then the Schrödinger equation in 1926, that many people began to understand that our assumption of understanding was flawed. And as a result, we made enormous progress with regard to things like electronics, and for that matter, cell phones and many other things. So the problem that we frequently have is that we think we understand something and we don't look any further. And this has really been true with regard to things in medicine too. Now, the real question then is, what are the underlying causes? And that's true of aging, but it's also true of many other things. Here is a sort of a trivial example historically, and that is plague. There are at least 3 clinical syndromes for a plague. There's bubonic plague, pneumonic plague, and septicemic plague. And if you knew nothing about the underlying cause, that is Yersinia pestis, the bacteria responsible for all 3, you would think of these as 3 slightly separate diseases that can overlap, but still appear to have different causes. And they don't. They have a common underlying cause that's expressed in different ways clinically because of the point of attack in the body. But the same thing really is true when we look at, for example, the age-related dementias. We know that there are clinical syndromes, for example, Alzheimer's dementia, Parkinson's dementia, frontotemporal dementia, in this case, vascular dementia. And these are real syndromes. We also recognize that there is some degree of overlap. For example, patients with Alzheimer's classically have cognitive decline, but can also have motor dysfunction. Patients with Parkinson's classically have motor dysfunction, but can also have cognitive decline. Likewise, patients with Alzheimer's or Parkinson's, when you see the brain at autopsy, you do find vascular dementia in a great many of these cases. And the same is true of frontotemporal dementias. There are overlaps. Now, that's not to deny the reality, in some sense, of the defined clinical syndromes, but just to point out, one, that there's a degree of overlap, and two, there is a suggestion that maybe there's an underlying cause going on. And the same is true when we expand these, look more carefully at things like Lewy body dementia or posterior cerebral atrophy, or any of the other alphabet soup of different age-related dementias. The consequence of this over the past decade or two is to talk about things like mixed dementias, because there's a clinical reality to that. Also atypical dementias, dementias that are age-related but don't truly fit into any neat category. And in fact, you find professional interest groups, you find actual global conferences on mixed dementias or atypical dementias. And the suspicion has been growing that these are all part of an underlying process of age-related neurodegenerative diseases that are expressed in different patients because of different genetic backgrounds, and for that matter, different epigenetic backgrounds. So for example, whether you've had head trauma or not, or infections or not, your genetic background, your diet, all of these, your exposure to herbicides, all these may have a different outcome in terms of the clinical syndrome that you get, whether it's Parkinson's, Alzheimer's, and so on. So that's kind of where we have stood recently. And this has brought to many of us this question, which is, when we talk about age-related dementias, do we truly understand aging? Because if not, how can we really understand age-related diseases? So again, we have to overcome our assumption of understanding, go a little deeper, and see if we can understand things. This was brought to a head in my mind about a year or more ago, when I was a part of a committee that organized a conference in Washington, D.C. on animal models in Alzheimer's disease. And I actually gave a summary talk at the end of the first day. The classic question, one that I addressed also, is why this occurs. Why is it that everything works in mice, but nothing works in humans? And in fact, not everything works in mice either. But classically what happens is we do an experiment, for example, with a genetically altered mouse using, for example, a human beta amyloid related gene. And when we can cure the mouse, we find that when we take it to FDA human trials, there's no effect. So the question is, why is that? And I pointed out that there are a number of reasons for that. One is that while we share about 75% of the protein expressing genes with mice, another 25% we don't. And almost invariably, the genes we look at with regard to Alzheimer's are genes we don't share with mice. So it's in some sense not surprising that when we undergo translational human trials, and we find that in fact the results from mice don't translate well to humans because we're using mechanisms that are not fundamental to both organisms. Now the other problem though is that we do not have a system, unified systems model. And that led to me publishing it. In fact, this paper came out as a result of that talk I gave. I was invited by the editors of Alzheimer's and Dementia, which was the organ of the Alzheimer's Association globally, to write up the model. And the result of that was that there were more than 600 reprint requests in the first 2 weeks. And what the model really says, though, is that we can define a unified systems model to explain age-related dementias, one that is consistent with all known clinical and other data, is predictively valid of the typical trials, for example, those of Eli Lilly or Biogen, Eisai, and offers an innovative and feasible point of intervention to see if we can intervene in human age-related diseases. Well, when I talk about it being a unified systems model, let me clarify a little bit. If we're talking about a unified model, we have to recognize that there are intermediate pathways that are not shared between different species. As I say, mice, for example, don't process beta amyloid the same way humans do. Now, there are underlying processes that are shared, and there are intermediate processes that are shared. For example, the way we deal with mitochondrial function. is very similar in mice and humans, but some other processes are not. Now, one of the fundamental processes we share with mice is the way we handle cell senescence, the fundamental processes of cell senescence, which we will come back to in detail. But if we're going to have a unified model, we need one that can explain cognitive decline in animals and also disparate cognitive decline in humans. Why is it that one patient of mine will get Alzheimer's, another patient will get Parkinson's, and yet they appear to be otherwise very similar patients? And the reason, I think, is, again, that we've got patients with different genetic backgrounds, different epigenetic backgrounds, different histories, and the result is we see different clinical syndromes. But there are fundamental processes in all human age-related diseases, as well as animal cognitive decline, that are shared. And those are the ones that we need to focus on. So a unified systems model has to be able to encompass and explain heterogeneity within dementias. So, for example, why is it that some patients will get Alzheimer's disease at age 50, others at age 80, others at age 100 still have no evidence of Alzheimer's dementia? So within any particular clinical syndrome, Parkinson's, Alzheimer's, Lewy body, and so on, why is it there are differences between people? Secondly, the heterogeneity between dementias. Why is it that one patient will have Parkinson's, another Lewy body, a third will have posterior cerebral atrophy? Why is it that the age-related dementias come in various shapes and flavors? Why are there different clinical syndromes? Also, as I say, the heterogeneity between species. Why is it that mice, dogs, cats, rhesus monkeys, gorillas, baboons, human beings all have slightly different courses when we look at age-related neurodegenerative changes? Also, upstream biomarkers or upstream mechanism. If we look upstream, what we find is that some patients, for example, will have two ApoE4 alleles, other two ApoE2 alleles, and yet in neither cases are those entirely predictive of the clinical outcome. Some patients will have a history of head trauma and therefore increased incidence of, for example, Parkinson's or Alzheimer's, and yet some patients will have one of those risk factors and yet not have the disease, or have the disease and not have the risk factor. The same is true of central nervous system diseases, infections, exposure to toxins, and so forth. So there are multiple upstream mechanisms, and yet they are not in any case totally predictive. Why is that? Likewise, downstream biomarkers. Why is it that most of our Alzheimer's patients show beta amyloid plaque, but not all? Why is it many more of them show tau tangles, but not all? Why is it we see differences in the way the alpha-synuclein is laid down in beta amyloid, in the substantia nigra in different patients? So there's a disparity of downstream biomarkers that we need to explain as well. And besides having a unified model that can explain these, we also need a model that is a systems model as opposed to a components model. Now, to explain what I mean, let me give you an analogy from engineering. Here you're looking at an Airbus 380 cockpit, and at the top you're seeing a Rolls-Royce Pearl 15 jet engine. In the lower right, you're looking at a Trent 900 turbofan and one of the fan blades in that turbofan engine. Now, I've talked to some of the people responsible for the design and maintenance of these machines, and they point out something interesting, which is that if that fan blade fails, if it cracks and comes apart in use, they do not simply ask what's wrong with the fan blade. The more logical question is to ask what's wrong with the fan blade in the way it works in the turbofan 900 Trent engine, in the Rolls-Royce Pearl 15 jet engine, in the Airbus 380 at 35,000 feet, going hundreds of miles an hour with a very high RPM and high temperature. In short, the question is, how is the system making that fan blade fail, not simply what's wrong with the fan blade? Well, the same is true of medicine. We need to be careful not to confuse the component with the system itself. So to give a typical example in medicine, this is the Ebola River in the Congo. And as you might guess, what you're looking at there is a list taken from a whole semester-long course I gave on Ebola disease. These are signs and symptoms of Ebola. I certainly could include fatality in that list. But these signs and symptoms are not the disease. These are signs and symptoms of the disease. They're components of the disease. They're biomarkers of the disease. They're things we can measure, but they're not the process. The process is a very complicated interaction between the Ebola virus and the human body, resulting in a cascade of pathology, the outcome of which are things like this: leukocytosis, a bleeding, diathesis. These are common outcomes, but they are not the disease. So we need to be careful to distinguish biomarkers from the system process. The same is true when we look at aging. For example, these are typical biomarkers at the macroscopic level for aging, things we're all well aware of. For example, the declining glomerular filtration rate, arterial disease, skin aging. These are things that we see in our patients, or we see in the mirror, or we see in those around us. as aging occurs, but they are not aging. They are biomarkers of aging, but they are not the process itself. The same is true when we look at the microscopic level. We can see declines in the rate of DNA repair. We can see methylation changes, telomere shortening. We can see declining in the ratio of adenosine triphosphate to reactive oxygen species, but these are not aging. These are components of aging. They're typical biomarkers of aging. They're things we can measure and should measure, but they're not the process itself. The same is true when we look at age-related neurodegenerative diseases. For example, beta amyloid plaque, tau tangles, changes in the blood-brain barrier, failure of that, lymphatic changes. These are all things we can see at the histologic level, for example, but they are not the disease. They are biomarkers, they're components, they're outcomes, they're signs, they're symptoms, they're not the process. And even if we go down a little further and look at the genetic level, these are typical of some of the dozens and dozens of gene predilections we find for age-related human neurodegenerative diseases, for example, Alzheimer's disease. But they're not Alzheimer's, they're not Parkinson's, they're not any of the age-related neurodegenerative diseases. They're simply genetic biomarkers of the disease, but they do not explain the process. To explain the process, we need to look a little further. So let me summarize this point about biomarkers by saying this. I think most of us recognize that if you can't measure something, it's not science. Very common statement. On the other hand, if you can't understand it, it's not science either. It is not sufficient to measure biomarkers, to have a whole list of signs and symptoms and components. That is not the same as understanding. And in fact, to go further than that, in medicine, if you can't understand it, then you can't cure it. If we need to be able to understand age-related disease, we need to understand the system, not simply the components. It's not simply a measure of measuring signs, symptoms, biomarkers of aging. What we need to do is understand the process itself at the most fundamental level, and as a result, be able to show we can intervene effectively and cure it. Well, 24 years ago, I gave the first lecture ever on this entire topic at the National Institutes of Health in Bethesda, Maryland, in the US. And I started that talk by making a point that I'd like to stress here, which is that anybody who thinks that you can reverse the aging process at any level is naive. Equally, anybody who thinks you cannot reverse the aging process at any level is naive. The question is, where's your data? And I proceeded to show what little data we had 24 years ago. The major point remains, what is the aging process? What is it, and what can we do to intervene at any significant fundamental level in the aging process itself? The result of that is I wrote a book on this some 24 years ago. I published the first papers ever on the prospects for reversing the aging process at the genetic and cellular level and the potential implications for human age-related diseases in the Journal of American Medical Association, as well as what was the first and still the only textbook on this entire area with Oxford University Press, with some 4,700 reference, quite a comprehensive academic book. Good luck to you. And a more recent book that the Wall Street Journal praised as one of the best science books of the year. And really what I was trying to do is address this fundamental question of what is aging and can we intervene? Now, let's step back and look at aging. First, from a species level. We know that human beings typically live, as I say, about 3/4 of a century. Gorillas about 40 years. That's Coco, the only gorilla who spoke sign language. I was actually her babysitter for about a year, 6 hours every Wednesday. I am humiliated to admit she had a bigger vocabulary in sign language than I did or do. Typically, domestic animals, domestic pets, for example, cats and dogs, live about 2 decades, depending on the particular breed involved. Typical rodents in the laboratory setting last about 2 years or a little more, again, depending on the breed involved. Now, most people would say that aging is simply a matter of entropy, which begs the question of, then why does entropy operate at different rates in different species? Most people then would say, well, it's a matter of genetics. People vary genetically from, for example, gorillas, dogs, cats, or mice, and that explains it. But it doesn't, any more than knowing the genes involved can really explain what's going on with Alzheimer's disease. We have to go a little further and not simply wave our hands and ascribe it all to genes, but dig down. And in fact, if we look down further within a species, we find the same sort of problems occur. And that is, these are all human beings, but these are human beings with progeria. These are 30 progeric children. Bill Sample in the back row with his hands up and I gathered them together every year from around the world. In this particular year, we took them on a tour of the White House in Washington, and we went places you generally don't go. I took hundreds of pictures of things you generally don't take pictures of. And we had a delightful time until Sammy, in the front row with the red baseball cap there on the left, and his friend Ori in the Hawaiian shirt beside him, inside that north portico door, sat on the third step up to the private quarters where there is a pressure-sensitive switch, which triggered the Secret Service to very politely but very firmly escort us out the door. Well, these children die overwhelmingly of age-related diseases. They typically die at age average of about 12.7. They are all bald. Those who appear to have hair are wearing wigs, with one exception who doesn't have progeria. It's also the reason they tend to wear baseball caps or shawls over their heads. They have very thin skin with prominent veins. Typically, they have osteoarthritis and osteoporosis, but they die overwhelmingly of age-related vascular disease, that is, strokes and heart attacks. There's a child in the back row here on the right who at age 7 later this same summer had a massive heart attack while throwing a baseball in the backyard with his father. Why do these children age so quickly? Well, we know the gene involved. It's the lamin A gene. But again, that's not an explanation. What we've done is identify a genetic biomarker, but we haven't explained how it worked and how aging works itself. So we need to dig a little further. We also need to point out that aging is not unavoidable. Every one of you is composed of trillions of cells, and every single cell in your body derives from a fertilized ovum, and that ovum you got from your mother, and at that time it was already a couple of decades old. She got it from her mother, who got it from her mother, and that line of cells can be traced back in an unbroken biological sequence for 4.5 billion years. And during the entire 4.5 billion years, those cells changed a great deal, but they never aged. The same is true of mitochondria. Every mitochondria in every cell in your body derived from your mother, who got it from her mother, and that line of cells in the eukaryotic line can be traced back about 1.7 billion years. And during that entire 1.7 billion years, those mitochondria never aged. And yet in both cases, the cellular sense and the sense of the mitochondria, there was an inflection point a few decades ago when you were born, and things began to change in many of your somatic cells. In fact, if I look at the mitochondrial function in your cells, most of them would show some age-related changes, which I'll come back to in detail later. But the idea that aging is avoidable or unavoidable is simply wrong. We know that there are cell systems and there are mitochondria that never show any evidence whatsoever of aging, and yet most of the common ones we're aware of in your body or those of domestic animals or other animals around show age-related changes. So we need to understand why that occurs. To do that, let me sort of simplify and say that aging is a balance. Clearly, entropy plays a role, but it's a balance between entropy and, in this case, maintenance. And what you see is that in cells that never show any aging, for example, the germ cell line that resulted in you, entropy is perfectly balanced against maintenance, but that process fails in many of your somatic cells. To use an analogy of gardens, let me say that the reason that some gardens last forever, these are gardens of mine actually, and they do very well, is not because there is no entropy, not because there are no weeds, it's because of maintenance, that is weeding. So any garden that survives because there is careful weeding to balance against the weeds. The same is true when we look at cellular systems in biology. So for example, if we look at the germ cell line, you find that entropy is exactly balanced against maintenance, and maintenance is maintained flawlessly for billions of years. But that is not true of many somatic cells. For example, the 80-year-old hand in the left there feeding the bluebird has dramatic changes in the cellular functions of the fibroblasts, the keratinocytes, the melanocytes, the hair cell follicles, almost all of the cells that are involved, including the arterial cells, and if we were to look at the brain, the glial cells. So the maintenance fails in aging cells. It is not a matter of aging being due to entropy. It's aging is due to a failure of maintenance in the face of entropy. So in young cells, what we have is long telomeres and a distinctive pattern of gene expression. Now, if I were to look at my young cells, any particular cell system, whether it was the keratinocytes, the podocytes in my liver, the glial cells in my brain, any of those cells have a characteristic pattern when I was, for example, 10 years old. Now, in those days, they had longer telomeres and that distinctive pattern of gene expression. Now, in my cells, as I've gotten older, I turned 70 in a month or two, the cellular expression of those genes is different. It's the same genes, but a different pattern of expression. In fact, throughout my body, almost all the genes are identical. Not quite, but significantly the same. The difference between old cells and young cells is not the genes, it's the pattern of gene expression. And this is modulated, as we'll see, by shortening of the telomeres. So to use an analogy, the difference between two symphony orchestras, one of which is playing Mozart and one of which is playing Beethoven or John Cage or anything, Glinka, is not necessarily the difference in the genes or the musicians. It is the difference in the score and the tempo. So I can have an orchestra that one evening is playing Mozart and the next evening is playing the Grateful Dead, and yet the instruments are the same. The difference between those two symphony orchestras is not that in one case the pianist has gone deaf, or the oboe has a broken reed. The difference is the score and the tempo set by the conductor. And the same thing is really true when we look at telomere function. The difference between old cells and young cells is not different musical instruments or different genes. It is the way that the score is changed in older cells. And if we look at this in more detail, what you see is that for any set of molecules, any molecular pool, for example, the collagen or the elastin between the cells in my skin here in my face, if I look at cells when I'm young, there is a rapid turnover of any one of those molecular pools. So for example, collagen. The collagen is produced. It may or may not be damaged. It is then recycled and created again. So you're continually having a recycling process or a turnover process in any molecular pool. And this is true whether we're looking at, for example, beta amyloid in my brain, or whether we're looking at chondroitin sulfate in my knee, or we're looking at elastin in my face and the skin of my face. There's a very rapid turnover, and the outcome of that is that the damage, signified here by the little red star, is washed out relatively quickly. So at any one time, when I'm looking for an elastin molecule or a cytochrome C oxidase or a catalase molecule, I'm likely to find very little in the way of damage and mostly effective molecules associated with longer telomeres, incidentally. As the telomeres shorten, though, they begin to change the pattern of gene expression throughout the rest of the chromosomes. And what you find is the molecular turnover slows down. And as a result, you begin to get a slight accumulation of increased damage, not because damage is increasing, the rate of damage is increasing, but because the turnover rate or the recycling rate of any set of molecules is decreasing. And in fact, in older cells, what you find is the turnover is slow enough that there is at equilibrium an accrual of damage significant enough to cause dysfunction to the cell. In fact, you can look at this mathematically. The formula is found here on the bottom. But what this really says is that if you have a rapid rate of turnover of any molecular pool, the incident damage at equilibrium is very low. Whereas if you have a very slow turnover, for example, in old cells, the incident damage at equilibrium is relatively high. So old cells tend to have ineffective and damaged molecules, resulting in an ineffective and cellular process or ineffective and damaged cells. You can actually show this mathematically also, because if you're looking at the recycling rate, as the recycling rate falls as you move toward the right, the percentage of damage goes up exponentially. And the same, by the way, is true of age-related disease. That is, as the recycling rate of any set of molecules goes down, your likelihood of having damage to cellular systems, tissue systems, organ systems, and in fact, clinical age-related disease goes up exponentially as well. Now, let's look at some examples of this in detail. The example I'm going to use here is from Alzheimer's disease, and we're looking at beta amyloid turnover. And what you see is that in young cells, there's a very rapid rate of production, secretion, binding, reuptake, and degradation of beta amyloid molecules. But as the cells, in this case the microglia, begin to get older, you find that that rate slows down a bit, and there's more time for the beta amyloid to form microaggregates. Now you might ask, what about the difference genetically between patients at high risk and low risk? For example, if I have two ApoE2 alleles, I have very low risk of having Alzheimer's. And it turns out that people with two ApoE2 alleles make molecules of beta amyloid that are relatively not sticky, so they have a low rate of damage. So even though the rate of turnover goes down, the rate of microaggregates is not very much higher, and so you get few beta amyloid microaggregates in these patients. Whereas if I have two ApoE4 alleles, I tend to make stickier molecules. And the result of that is I have a higher rate of damage, so I see many more in the way of beta amyloid microaggregates, even at a younger age. But as we all get older and that rate of turnover gets even slower, we tend to see an increased incidence of beta amyloid plaque, almost regardless of genetic background. Clearly more with with ApoE4 alleles than with ApoE2 alleles, but all of us tend to have more in the way of microaggregates and amyloid plaque. And the same is true when we're looking at other biomarkers of age-related neurodegenerative diseases. For example, tau tangles, mitochondrial dysfunction, which we'll come back to, inflammatory changes, and so on. And the result of that is that you begin to see synaptic loss, dysfunctional cells, dead neurons, and clinically speaking, you see age-related neurodegenerative diseases or clinical dementia, in this case, Alzheimer's, but just as true of other age-related dementias. Now, I said we'd talk a little bit about mitochondria because they change dramatically with aging as well. 99% of the aerobic enzymes that you use in mitochondria originate from the nucleus. And as the nucleus slows down its rate of turnover, that is production and degradation of these aerobic enzymes, you begin to find that the enzymes used in your mitochondria are less and less efficient as your cells get older. And the result of that is you make less and less in the way of adenosine triphosphate, the essential power molecule for your cell, and more in the way of reactive oxygen species, that is free radicals. That is, your aerobic processes become sloppy. Nor is that all. The lipid membrane that surrounds mitochondria, it becomes leakier. And the reason is that the lipid molecules are also being actively turned over. And that active turnover slows with age as well. So not only are you making more in the way of free radicals, but you're leaking them into the cytoplasm, and the nucleus for that matter, more rapidly than you normally would. Moreover, you have scavenger molecules, for example, superoxide dismutase, catalase, and so on, that are responsible for grabbing those free radicals and essentially detoxifying them. But also, the inherent molecules you make in your cells, as opposed to the dietary ones, are slowed down in their rate of turnover as well. So scavenging rate goes down. So you're making more free radicals, you're leaking them into the cytoplasm, you are slower at scavenging them, and when you cause molecular damage, you're slower at turning over that damage. Nor is that all. Almost all the molecules in your body are not repaired. They're simply recycled. That is, whether you have a damaged cytochrome C oxidase or not, your body still tends to recycle it more slowly in older cells than younger cells, but you recycle it. However, there's one set of molecules that you actually repair, and those are DNA molecules. While you've been listening to me for the last 35 minutes, almost every single cell in your body has shown evidence of DNA damage. And almost without any error, you have repaired all of that DNA damage. You have 4 families of enzymes responsible for this. That is detection, excision, replacement, and ligation. All 4 of those slow down as the cell begins to show age-related changes. And the result is your ability to detect, excise, replace, and ligate DNA damage goes down exponentially, since each of these are independent processes and they multiply. So the outcome of that is that with age, you find this exponential increase in malignancy. And this is true whether you're a 2-year-old mouse or a 100-year-old human. The rate goes up exponentially regardless of the years. The defining characteristic is the aging of cells, not the aging of the calendar. You might in fact say that most cancer is not caused by your lifetime exposure to carcinogens, but your lifetime decrease in your ability to repair DNA damage as a result of carcinogens. Now, let me go back to telomeres for a second and point out that telomeres in some sense are irrelevant. The question isn't, do telomeres cause aging? Because they certainly do not, any more than genes cause evolution. Genes are a key player in the evolutionary process, but they don't cause it. Likewise, telomeres don't cause aging, but they are a key player in the aging process at cellular level. In fact, the entire concept of causation is a little sloppy and in some sense naive. It implies sort of a linear causation, A causes B, which causes C. And the entire process, whether you're looking at evolution or at aging, is far more complicated than that. Much more complicated systems process, a dynamic systems process, than a linear process. Furthermore, telomere length is irrelevant. It turns out that telomere length doesn't matter. What does matter is the rate at which you lose telomeres. So for example, you can predict the lifespan of almost any species, not by looking at telomere length, but by looking at the rate of loss. So for example, mice lose telomere lengths very rapidly, humans lose telomere length very slowly, and the result is that we have longer lifespans than mice. To give you a clear idea of what I mean by telomere length not mattering, let me point out that there are types of mice that have telomere lengths that at birth are 10 times longer than mine, but I have a lifespan 40 times longer than that variety of mice. So the length of the telomere is irrelevant. What matters is the way that length changes over time. You might say absolute length doesn't matter, relative length does. Moreover, it's not the telomere that matters so much as the change in gene expression that's incumbent upon it and the effects it has on molecular turnover at all levels. This is a way of looking at that complex process, taken from that paper that I published earlier that year that got so much global acclaim. Really what this says is that there are upstream factors that affect aging in cells. There are downstream biomarkers associated with those aging in cells. And in between, there's a cell aging process itself. So for example, we know that certain genes can increase your rate of, in fact, cell aging and cell damage. For example, ApoE4 alleles in Alzheimer's. We know that chronologic age plays a role. Exposure to various toxins can play a role. For example, exposure to paraquat, an herbicide, can increase your risk of Parkinson's disease. We know that trauma, head trauma, can increase your risk, whether it's trauma to the knee, increasing your risk of osteoarthritis, or the risk of head trauma, increasing your risk of a number of age-related dementias. We know that hypertension and hyperglycemia can increase your risk of age-related vascular changes, including age-related vascular dementias. The same is true of a number of other things. Radiation to the skin, for example, ultraviolet increases skin aging. The history of various infections, whether it's viral infections, bacterial infections, chlamydial infections, fungal infections, or prion infections, can increase your risk of cell aging. And the result of that is cell division, shortened telomeres, altered patterns of gene expression, cellular dysfunction, and the outcome of that is a number of biomarkers. Now, although this looks complex, let me point out that none of this matters to me. The question from my perspective is not what causes aging, but what can we do about it? That is, where's the single most effective point of intervention clinically if we want to deal with age-related diseases? And the answer is that the upstream risk factors are not efficient targets. Example, we know that if I can give you good glycemic control and good blood pressure control, I can lower your risk of age-related vascular disease, but I don't bring it to zero. We know that if I make genetic alterations, likewise, I don't bring your risk to zero. If I lower your risk of head trauma or your history of head trauma, I don't totally prevent age-related dementias. No matter what I do upstream, and these are worthwhile targets, they're not efficient targets. They're targets we've gone after medically now for centuries, but they're not targets that actually prevent age-related diseases. We likewise know that the downstream targets are not effective. So for example, if I go after reactive oxygen species, we find it does not have a significant impact on preventing or curing age-related diseases. The same is true when we look at some things like beta amyloid or tau tangles. There in fact have been more than 1,100 interventional trials on Alzheimer's disease, none of which by global consensus has shown any effect on the disease course. So we need a more efficient target. And that more efficient target lies at the center of this system, namely the aging process in cells itself. And the reason I say that that is the optimal target is because it works. In fact, this was first demonstrated 22 years ago, looking at the use of telomerase to reset the pattern of gene expression in human dermal cells. This is an expression from the Bodnar et al. Paper in Science in 1998, in which that group showed, the group from Jaron, showed that in fact, you can change the senescent pattern of gene expression and return it to that of a young pattern of gene expression in human cells in vitro. You know, 2 years later, the same thing was done by Walter Funk and his colleagues again at Jaron, in showing that if you take aged skin, you can actually turn it back to young human skin in the laboratory, again, with no more change than using telomerase to reverse aging in human tissue in the laboratory. And the same is true when you're looking at human bone, or whether you're looking at human vascular tissue. You can actually reverse aging in human cells and human tissues in the laboratory. So the question is, can we do that in organisms? And there's a fair amount of data that's increased over the past decade or two, suggesting that we can if we can find a way to do it effectively. This is a paper from De Jesus and the lab with Maria Blasco in Madrid at the CNIO Institute, in which they showed that in fact you can use telomerase to reset cell aging, neural function, and in fact reverse cognitive decline in animals in vivo. It works fairly well within technical limits. The result of that is that we're actually going to take this to FDA human trials and show that we can prevent and reverse cognitive decline and age-related dementias in human beings. This is our scientific advisory board. Zavin Khachaturian over on the left there is the editor-in-chief for Alzheimer's and dementia. Suzanne Hendricks, for example, has been responsible for a lot of the design of Alzheimer's trials globally for large pharmaceutical firms. Richard Mose is the chief scientist for the Global Alzheimer's Platform, and so on. Our clinical advisory board includes people like Steve Arnold and Lon Schneider from Harvard and Keck USC, as well as Russ Swerdlow from KUMC, where he's the director of the Alzheimer's Center. These are people who have global credibility and are very careful, thoughtful people. They are going to make sure that we retest this model accurately and rigorously in clinical trials. We have actually already talked to CBER, the Center for Biologics Evaluation Research at FDA. We will be using a single-dose gene therapy as an intervention, using an AAV virus, AV9, and a human telomerase plasmid, targeting glial cells and neurons in the human brain. We intend to show that we can actually reverse the cognitive decline seen in age-related dementias, starting with Alzheimer's and progressing on to other age-related dementias, including Parkinson's disease. We think we can actually go beyond this and show efficacy in treating other age-related diseases like age-related vascular disease, osteoarthritis, osteoporosis, age-related changes in glomerular function, and we will see where this progresses over time. I'd like to thank all of you for joining me in this conversation today. For those of you who have questions, please feel free to get in touch with me. I look forward to talking with you. Good luck to you, and let's see what we can do regarding to aging and age-related diseases. Thank you.